I do know her. I've met her before. This is Manshi Zhou, and she's a grad student at the University of Glasgow. I got it right. Okay.
Hello. Am I on the mic or okay? So
You want to do a TED talk?
I think I'll just be here. Um is it on? Okay. So I will be presenting my renal multi-omic analysis of UMOD knockout mouse in response to salt. So previously, our lab did a genome-wide association study on human population, comparing the blood pressure. I think I don't know if the people online can see it. I'll just use the mouse pointer. And well, previously our lab did a GWAS on human population, comparing the blood pressure extremes, and identify SNP RS13333226 at the promoter region of UMOD, uromodulin, with a minus G allele associated with lower risk of hypertension and lower level of urinary uromodulin. Just to point out that UMOD here is on the reverse strand. That's why the SNP here is at the promoter region. Uromodulin, otherwise known as tamhorsoptotide, is the most abundant urinary protein and is almost exclusively produced at the epithelial cells lining the thick ascending limb at the loop of Henle in the kidney. Its potential roles include immune function and calcium signaling, and it's known associated with chronic kidney diseases. This is our blood pressure validation in the UMOD knockout mouse. Here is the telemetry data. We measure the blood pressure without any intervention starting from the age of 12 weeks. We can see this dent here. It shows the UMOD knockout mouse. It's significantly lower in systolic blood pressure than the wild type. And then we add the salts to their drinking water for 6 weeks, again, starting from the age of 12 weeks. We can see the top graph tail cuff. In the wild type, it's significantly increasing the blood pressure after salt loading. However, in the knockout, we don't see any changes in the blood pressure. So the blood pressure is insensitive to salt loading in the knockout, which is a very interesting phenotype. So we have the hypothesis of variations in UMOD expression level modify the mechanisms of blood pressure regulation, which led to our two aims: bulk RNA-seq analysis to identify potential genes, transcripts, and the related pathways that are associated with changes in blood pressure, and proteomic and metabolomic profiling to characterize the molecules that interact with uromodulin. So today I would just focus on the RNA-seq analysis and proteome analysis. This is our experimental design. We have the strain comparison knockout versus wild type, and also the salt comparison high salt versus normal salt. We have N of 3 per group, and all of them are male. We took the whole kidney, grounded and extracted the RNA, protein, and metabolite from the same ground tissue for parallel bulk RNA-seq, 16-plex TMT, proteomic, and untargeted metabolomic analysis. Well, thanks to the two previous speakers, you have a bit of exposure of the workflow of the RNA, the proteome and the metabolome workflow. So I wouldn't talk anything about the workflow. And this is our RNA-seq data with a cutoff FDR less than 0.05. We only, this is unexpected to see only 4 differentially expressed genes in the strain comparison under the high salt condition, considering our very distinct blood pressure phenotype that the knockout is insensitive to salts, but the wild type it's significantly increased upon salt loading, which we just need further explore. And today I will just focus on the salt comparison within the wild type and the salt comparison within the knockout. We can see only 700 differentially expressed gene in the wild type, but 6 times more in the knockout. So when we look at the volcano plot, we can see the top here is the wild type, and this evenly distributed pattern in the wild type. However, when we look at the knockout, we can see a negative skew, which means salt loading actually switch off many genes in the knockout. And then we use ingenuity pathway analysis to look at overrepresented and significantly regulated pathways. And each bubble shows a canonical pathway. We can see only 4 in the wild type, but much more in the knockout. And the blue means it's downregulated, so we can see a lot of the pathways downregulated in the knockout. Particularly, unfolded protein response is the most significantly downregulated and is the most overrepresented pathway in the knockout data, which I will come back later. Let's look at the aldosterone signaling first. This is just the portion that includes only the ion transport related genes, and the top figure is overlay with our wild type data, and the bottom overlay with the knockout data. We can see not much changes in the wild type, but the potassium and sodium related sodium potassium transporter related pathways, they are significantly differentiated in the knockout. So we did the disease and function analysis to specifically look for transport of molecules. And we saw that 68, that's unique to our wild type sort comparison, shows it has it predicts to activate the transport of molecules. And in contrast, when we look at the knockout, much more of them are unique to the knockout dataset, and they predict to downregulate transport of molecules. And particularly, it significantly downregulates the transport of protein, which includes protein trafficking. Next, we look at the unfolded protein response, which I mentioned earlier. This is the entire layout of the unfolded protein response canonical pathway. And this is a overlay with our wild type data. We can see only 1 gene that's downregulated. The rest of them doesn't show any changes. And in contrast, when we overlay with the knockout, we can see almost all the genes in the pathway are downregulated, with the green means downregulated and the red is upregulation. And then we selected HSPA5, DNAJB1, and also HSPA1A for TAPMAN validation. HSPA5, otherwise known as BIP, it's a biomarker for the UPR pathway. And we can see here it's significantly lower expression in the knockout high salt than the knockout normal salt. And although we don't see statistical significance in the other 2 genes selected, we can see this downward trend in the knockout high salt versus knockout normal salt. So it would just be the data in the normal salt. It's a bit noisy. We would just need to increase our sample size. Then we look at our protein data. So these are the significant cutoff with an additional log fold change higher than 1.2. And as you might recall from the RNA-seq data, we only have 4. Here shows 346, which gives us a little bit more information to look at the strain comparison in the under the high salt condition. But today, just to be consistent with our RNA-seq data, I will just focus on the salt comparison in the wild type and the knockout. And we can see almost double differentially regulated protein in the knockout than the wild type. This is the ingenuity pathway analysis, and I selected the top 10 most significantly overrepresented canonical pathways. The top figure is overlaid with our wild type data, and the bottom is the knockout. And we can see mitochondrial dysfunction is significantly upregulated in the wild type, and the oxidative phosphorylation is significantly downregulated in the wild type, which we observe a opposite direction in the knockout. So the mitochondrial dysfunction is downregulated, and the oxidative phosphorylation is upregulated in the knockout, which shows the two strains respond differently in energy production related pathways. Just to have a quick glimpse and of how the protein spread out in each of the pathway. This is the mitochondrial dysfunction. We can see the proteins that's identified in our data set. They are at different stages of the pathway, and also it shows generally upregulated in mitochondrial dysfunction, and with a z-score equals to 3.2, which means it's significant when it's higher than 2. And in contrast, in our knockout, we can see it's generally green, which is downregulated. Again, in the oxidative phosphorylation, we can see downregulation in the wild type, but upregulation in the knockout. In summary, consistent with our blood pressure phenotype, RNA-seq suggests UMOD's potential function in modulating ion transport, protein trafficking, and stress response. And our proteome data alludes to opposite effects in energy production between the two strains, specifically mitochondrial dysfunction and oxidative phosphorylation. In conclusion, uromodulin is a multifunctional protein contributing to intracellular and intercellular transports and signaling. Yeah, thanks to my supervisor, Dr. McBride and Dr. Graham, and the lab members and the team. And just to mention that Kara will have her speech on Thursday, so you can hear about a different project in our lab. Any questions?